Hello, everyone, and welcome to the kickoff of our fall portion of our 2024-2025 Phi Tau Sigma webinar series. The purpose of these webinars is to provide scientific and professional development programming to our membership and potential new members. The live programming that you see today will be available to all, but then the recording will be available on the Phi Tau Sigma website for the membership. My name is Elizabeth Grasso-Kelly, and I am the current president of Phi Tau Sigma, taking over for Dr. Terry Boylestone, who just finished up her term at the end of August. Last year, I was the program committee chair. This year, it's Dr. Finman Kong, who is going to be leading the effort. So we'll be working together on the remainder of this webinar series. I'll also be serving as today's moderator. So if you have any questions or comments as we move forward, please just reach out to me. If you have any questions for our speaker today, you can put them into the chat and I can ask them at the end, or you can come off mute at the end of the presentation and ask your question. Today, I would like to recognize Dr. Ali Ubeytogullari. Sorry, apologies, Dr. Ali. He is our 2023-2024 Early Career Accomplishment Awardee. So he is the recipient of this award. This award is really designed to recognize outstanding contributions in the field of food science and technology by Phi Tau Sigma members early in their careers. This award comes with a plaque, an honorarium, and then the ability to provide a presentation to our membership that you're seeing here today. Dr. Ali is a lifetime member of Phi Tau Sigma. He received his PhD in food science and technology from the University of Lincoln, Nebraska. And then he went on to Cornell University for his postdoctoral research. Currently, he is an assistant professor in the Department of Food Science in the Department of Biological and Agricultural Engineering at the University of Arkansas Division of Agriculture. Dr. Ali directs the Food Engineering for Health Lab, which focuses on enhancing human health and wellness through food using sustainable, innovative food processing technologies. His current research interests include enhancing the bioavailability of bioactive compounds using supercritical carbon dioxide technology, nanoscale science and engineering, and 3D food printing. Today, Dr. Ali will be speaking to us about a novel 3D food printing approach to generate micronutrient delivery systems. So at this time, I'm going to stop sharing my screen so he can start sharing his and we can proceed with the presentation.
Perfect. Thank you, Elizabeth. Can you see the screen in full size?
It looks correct. Thank you.
Perfect. Yeah. Thank you so much for the introduction. And I am happy to give this first lecture on novel food printing applications to generate micronutrient delivery systems. So my name is Ali Ubeytogullari, and I am assistant professor in the Department of Food Science and the Department of Bio and Ag Engineering at the University of Arkansas. So before I start, I would like to thank the sponsors of Phi Tau Sigma Early Career Accomplishment Awards, Dr. Lauren Jackson and Phi Tau Sigma. It's a great honor to be the recipient of the first recipient of this award. So as Elizabeth mentioned, so I got my BS and master's from Department of Food Engineering from Middle East Technical University in Turkey. And then I moved to Lincoln, Nebraska for my PhD, where I worked really with supercritical carbon dioxide to create aerogels and do really different things with CO2 on extraction, reaction. And then I moved to Ithaca, New York, and at Cornell, I worked with Dr. Rizvi. So we, again, did more research on CO2. So I was involved with multiple research on fractionation of phospholipids from milk fat, as well as we did some extrusion with the help of CO2. So in my current lab at the University of Arkansas, we are really looking at the intersection of food engineering and human health. So on the engineering side, we really use novel processing technologies like supercritical carbon dioxide, nanoscale science and engineering, And on the other side, we look at the digestion and bioavailability to see what happens to those nutrients after they go through processing. We look into nutrient delivery, and we combine those two using additive manufacturing, and specifically we use 3D printing here with the help of material science. So first, let me give you an overview of my research program here. So we do some research on upcycling food processing byproducts, and some of those are rice husk, rice bran. As you know, rice is a big in Arkansas. We produce more than 50% of the rice in the US, and those are some byproducts from that processing. We also did some research on grape pomace. We work on sorghum bran, and we try to upcycle those and create value-added materials out of those underutilized materials. Of course, we do a lot of extraction of bioactive compounds. We use supercritical carbon dioxide for that purpose. We also do fractionation. We don't have really a specific commodity. We work with different underutilized, or even just to enhance the extraction process. Sometimes we use CO2 as alternative green technology. Another area we look into is aroma enhancement. There are a lot of problems with off flavors and such, so we are looking into clean processing technologies such as supercritical carbon dioxide to enhance the aroma of, in this case, we did with sorghum flour, and we try to remove off flavoring compounds, volatile compounds. So on the other side, we work with 3D printing, and first area is, I call it macro scale. So basically in this, where I'm talking more on large scale printing, where you can generate snacks or cookies or more like products that are finished and ready to consume. And then we have another area where we look into encapsulation, where we do more micro printing, where we are looking into microstructure, and we are trying to encapsulate bioactive compounds. So before I move on 3D printing, I will shortly discuss what we do with CO2 with few examples on supercritical carbon dioxide in our lab. So what is supercritical carbon dioxide? It's a green alternative technology that we use really for different purposes. So I have done a lot of things with CO2. That's my background, and probably I have used CO2 for almost all the potential areas that commonly we see in the literature, from really extraction, particle formation, aerogel formation, reactions, separation, fractionation, reaction we did. Then we did also extrusion. So we really use it for different purposes, and just by definition, it's basically CO2 that is above it is critical temperature and pressure in this region, and we can play with the properties of CO2 with changing temperature and pressure, so that we have a solvent that can work for a specific application. So why we are using CO2 as a fluid? So here, CO2 is non-toxic, inexpensive and abundant, and it's an environmentally friendly process. You don't really need high temperature or pressure to reach that supercritical region, and you can easily separate CO2 at the end of the process. And since you are using pure CO2, we do not really have oxidation, and you can clean label your products since you're not using any petroleum-based toxic solvents. So just some applications that we are doing recently in the lab. So one application we did trying to separate high-value lipids and phenolic compounds from sorghum bran, and here we use a sequential CO2 extraction process where we first able to get the pure with pure CO2 the wax-rich fraction, which is the lipids, and then we modified CO2 with the use of ethanol, and we are able to get hydrophilic compounds. So we can do both hydrophobic and hydrophobic with this system, and we characterize the extracts here. We have different phenolic acids, but also we have 3-deoxy anthocyanidins that are unique to sorghum. And the other promising application we are looking into here, this wax-rich fraction, we looked under the ESC, and we are seeing really high melting point waxes that can potentially replace carnauba wax in the industry. So after we got those results, especially about the wax fraction, we looked at another side stream that is from bioethanol production. Basically, they are ending up with lipid slurry, and that's really underutilized. And we tried to look into this because sorghum is also used for fermentation, and then you end up with this side stream. And since we know sorghum bran has high value wax compounds, we looked into this underutilized source, and we tried to fractionate this material using supercritical carbon dioxide. So here you see the initial slurry, how it looks, and then we are able to really get more purified oil on one side, and we have the waxes on the other side. We are still working on this process, and even we are able to get powder wax fraction from this. So we used our SM design to optimize the protocol, and here you see the melting curves or DSC curves of those samples. And as you would expect, we have higher melting peaks here, but of course this needs to be further purified so that if you want to get really single peak at the high melting conditions like 85 or so degrees Celsius. So, and another application we did with CO2 is to create aerogels, and I have been doing this for a while now. So here we looked into defatted rice bran. So what happens in the rice processing industry? They remove the bran, and then you generally do an extraction to remove the rice bran oil, and then you end up with this defatted rice bran, which is really underutilized. And we looked into this, how can we upcycle it and create high value materials? And in this case, we created aerogels with really a high surface area and porosity that can be used for delivery of micronutrients. And again, with aerogels, we really use different materials here. And here you are seeing some images from cellulose and starch mixtures that are used for delivery systems. And we had the one review on this. And that was the cover of the journal, which is one of the IFD's journals. So that's the background about CO2 so far. And now I would like to move on to our topic today, which is 3D printing. And here's the outline. First, I will talk about the fabrication of beads, particles using 3D printing. Here we will look into starch and allogeneic pectin as two materials. And then we will discuss the encapsulation of lutein in starch matrix using 3D printing. And here we will look into two different core materials to solubilize lutein. One is ethyl cellulose and the other one is zane. And then we have the third part. I will briefly discuss some applications on reducing food waste by upcycling imperfect vegetables using 3D printing. So first of all, why we are doing this, right? And as you may know, bioactive compounds have several health benefits, and these include curcumin, lutein, phytosterols, other carotenoids, tocopherols. You really can, list a lot of bioactive compounds with numerous health benefits. But the problem is their chemical stability most of the time is very low, so they are not stable and they degrade easily. And another problem is their absorption in the body is very low, which causing to low bioavailability. And those two things really prevents their application in foods. And at the end of the day, we waste those materials, even though they are really expensive food ingredients that takes a lot of energy to obtain. So what we are proposing here is to use 3D food printing to encapsulate those compounds to increase their chemical stability as well as their bioavailability in the body once consumed. So one of the polymers we are using here is starch. and why we are using starch. Starch is inexpensive and abundant. Really, it presents in many food formulations, and so that you can easily incorporate that into foods. Another advantage is it creates a porous network without really using any crosslinker or chemicals. You can just heat that up and create a gel which has a porous structure, which can really protect and deliver bioactive compounds. So before we start on how we are using 3D printing to create those particles, first let's look at what's the conventional way that we have been using to create hydrogel beads, especially from starch. So the common method is called the emulsion gelatinization method. And what you do basically here, you create a polymer dispersion. So you create starch in water, and then you emulsify that. You use oil and emulsifiers, and eventually we heat the system up so that the starch dispersion gets gelled, where you have kind of a droplets hydrogels, and then you do centrifugation to separate the particles. And eventually what you need to do is you need to extract the oil back using generally a petroleum-based solvent, which is hexane. So there are a lot of disadvantages, limitations with this protocol. You really need a lot of oil, emulsifiers, and then you need to use an organic solvent to be able to extract them back. There are a lot of extraction, purification steps that goes into this process, which requires a lot of energy. And another limitation, you do not really have control where you locate your bioactive compound, and there are issues with the scale up because of that. So our proposed approach is to eliminate really the use of oils, emulsifiers, and toxic solvents in the process to generate starch particles or beads using 3D food printing. So some advantages that we see with 3D printing, you can really, one of the promises here is to create personalized food. You can really customize ingredients, use alternative ingredients with minimal waste. One thing that I think is important for especially our studies, 3D printing provides you high flexibility and precision. So you can do different things using the same system, but you can do it precisely, meaning that you really have control over the structure. So you can control where your bioactive compound is located at during printing in your food formulation. So in the first study I will share, we looked into extrusion-based 3D printer, and here we load basically the starch paste in a cartridge, and we dried the particles using freeze drying, and then we looked also into supercritical carbon dioxide drying. So in terms of 3D printing parameters, we looked into nozzle size. So we used different nozzle sizes here, different starch concentrations. Then extrusion temperature was constant, which was due to looking into starch gelatinization. And then we used different pressures for extrusion. And here you see some of the images, of the particles that we obtained at different nozzle sizes and different starch concentrations here. So we will talk more into the properties, but you can easily tell as you go from here to here, the particle size is going down. But we also quantify that using ImageJ software, where we look into characterization of the particle size. So one thing that's very important when you do 3D food printing is the rheology. So the rheology really dictates how stable your print is, or what concentrations you need to use to obtain something that has a good shape fidelity. And that's why we really look into rheological properties when we do 3D printing. So one thing here that we look, and probably is important to obtain a good 3D printing, is shear thinning behavior, so that as you apply more shear during printing, which is basically you have a cartridge and then it goes through a nozzle, right? So the pressure or the stress at the nozzle is higher than the regular material you have. So as you push it through, you are applying more force, and as you apply more force, you want your viscosity to go down so that it's easier to extrude. But another thing that we will discuss in the next few slides is how it bounces back, which is the recovery part. But that's one thing we look into: shear thinning behavior. We also look into storage and loss moduli to see yield stress and compare those. Tangent delta here, we look into that to see how and the gel behaves. And in this case, we have an elastic gel that can support 3D printing. So from those images, we really collected the data on mean particle size and sphericity of those particles. And here we have the starch concentration with different nozzle sizes. So looking at the particle size, we obtained the smallest particle around 650 micron with the smallest nozzle size, as you can expect, with the high starch concentration, which was due to improved gel strength, and it was able to hold the structure better. For sphericity, at that condition, we have around 0.87, which is okay for spherical particle with 3D printing. We did obtain higher sphericity for some cases, but the particle size was larger. So in this case, we chose those two conditions as the best for starch beads. So looking into their bulk density, so you see here their densities went up as we are using more starch, which is basically you have more material in the hydrogel, and that's why it's higher. And on the other side, the porosity decreased with the increasing the material, as you have more solid material in the mix that replace the pores. But overall, looking at the numbers, they were all about 84% porosity. So they were all porous beads. And we can see that looking under the microscope. So here you see some SEM images of the surface. And then we are seeing the center here. So they are all porous, and those are all freeze-dried starch beads. So we do see the thickness of the shell on the surface changes with increasing the concentration, but mainly this non-porous surface was resulting due to the drying process, but also it can maybe enhance the delivery and the protection of some bioactive compounds loaded in the beads. So after we did that study, we looked into different amylose content starch. So we looked here, you see corn starches with 72 amylose content, 55 and 25, the regular corn starch. So we looked into those and we compared their 3D printability, as well as we looked into different drying methods. One is the freeze drying, and then the other one is CO2 drying. So here you see the recovery test we do. And as I mentioned before, when you apply pressure through a cartridge, and what happens, the material goes through the nozzle, and as it goes through the nozzle, the pressure or the stress on the nozzle or the material increases. But then once it is deposited, those forces are released, and then the material needs to recover back. And depending on how much it recovers back, the stability of that layer changes, because you are going to add the next layer on top of it. And if that first layer is not stable, you will not be able to build that 3D shape. And recovery is one of those tests that we use a rheometer. We apply, increases shear rate. First, we use really low shear rate. So in this case, I think we went with 1 per second, and then you do 100, where the viscosity goes down, and then again you reduce the shear rate so that the viscosity bounces back, and that dictates really stability of those eventually once you print them. So those are the beads we created, and here you are seeing the images of the particles as well as their inner structure under SEM. And those are the ones dried with CO2, and they are called aerogels. So if you do the same process and dry it with freeze drying, then it's called aerogel, cryogel, sorry. And here are the images of those. But one thing to compare here, so this image here has 1 micron scale bar and a lot of porosity, right? On the other hand, when you do freeze drying, this is the same scale bar. We do not see any pores in any of those insets, even though we still have pores, macro, large pores, but the microstructure really is lost during drying. And we will come into that. We really see that in the surface area data. So the bead size we characterized here for those different starch types, and the aerogels were smaller than the cryogels, which was due to the drying process mainly. And with hydrogel was pretty similar to the cryogel part. And their sphericity for both drying methods were really similar. But the biggest difference is here. So in this table, we have the properties of the aerogels. So here you see the surface area, pore size, and pore volume. So when we use higher amylose content, we obtain really higher surface area. And this was around 175 square meter per gram with around 14 nanometer pore size, 0.7 cubic meter per gram pore volume. So now if we compare this with the cryogels are really low in terms of surface area. So the equipment was not able to even measure. So the report we got was less than 1 square meter per gram for the cryogels. So there is a huge difference in terms of their surface area just because of the way of the drying we do. And CO2 is known to be one of the best drying methods to eliminate surface tension and capillary forces during drying, which really protects the structure. And then we looked into, X-ray to see the crystallinity, and this is not really due to printing or drying. It's more like about gelatinization part where the starch, amylose swells and leaches amylose. So we see a decrease in crystallinity because of the gelatinization. So next we looked into, again, crate particles, but this time we are looking to alginate pectin. This is more for pH sensitive delivery of systems. And the conventional method is basically you add dropwise alginate solution into a curing solution. This can be calcium chloride with a peristaltic pump. And the limitations here, you don't have consistency. Pump calibration can be issue. And also you are not able to really encapsulate compounds since you just generally mix the two ingredients. So again, we are using 3D printing here with high precision and flexibility to create those beads. And we looked at different gum concentrations from 1.8 to 2.2, different nozzle sizes and extrusion pressures. And again, we looked at the rheology. So looking at the images, so here you see the peristaltic pump images at different nozzle sizes, and then we compared those with 3D food printing. This was the smallest nozzle size we used. We were not able to generate one with the peristaltic pump, but we are able to create those particles still with a 3D printer, and we see clearly the reduction in size by using a 3D printer, and also we have higher precision, and that can be observed from the smaller error bars. So far we talked about the particles, and now I would like to move on encapsulation of actual bioactive compounds into starch matrix. And here we are using a different design with the coaxial nozzle design. Basically, we have the core with lutein involved, and in this case, EC represents the ethyl cellulose, and then we have the shell with starch paste. This is the geometry we used, this cubic shape. And the idea here is, as you load those, basically what happens, the center loaded with lutein is dissolved in ethanol, and that diffuse into the aqueous starch solution. And as it mixes together, the solubility of lutein decreases and they precipitate when drying. So the idea is those materials will prevent the formation of large crystals and reduce their crystallinity. So the design really we use, so here you see an image that I took from a local restaurant here, Cosmos Cafe. It's a Greek cafe. And basically this shape you see on this image, this one is called Greek key. And historically it has provided enhanced structural stability. So we looked into different starch concentrations. We went from 9 to 11. We looked at ethyl cellulose concentration, and we looked at starch printing temperature. So again, the rheology, we are seeing all shear thinning materials for the starch different concentrations. Ethyl cellulose does not show a strong gel, as you see, and the viscosity is much lower than starch. But what happens basically during printing, we are using coaxial nozzle, and as ethyl cellulose solution interacts with the aqueous part, ethyl cellulose gets more solidified and able to hold the structure. And then we use some mathematical calculations here to determine the shear stress during printing for the inner and the outer shell. And here is some of the data. So in this case, we have two, coaxial nozzle, and we looked into the shear stress here as well as in the second one. We did this one because we wanted to know what's the shear stress in the nozzle so that we can do our recovery test based on the material instead of just choosing random one high and low shear rate values, so that we are able to get those numbers from here and use them for rheological analysis. This is just the image, micro CT image of the shape that we printed in this example. Another parameter that we looked into is the layer height. Layer height is really important when you do 3D printing because it affects, as you see in those images, it affects the overall shape fidelity. So meaning that if you have the first layer and your layer height is not high enough to go above that layer, you are basically printing on the same layer again, which distracts the gel, as you see in this image. But then as we increase the layer height, if it's too high, then you have those kind of gaps in between. And the bottom images are the micro CT images, and we can see the cross section of those gels so that the black spots you see on the image shows the gaps. And this one, we don't really see the structure clearly. So the structure was significantly improved in this. But again, in this one, we have a lot of pores, which negatively affects the shape fidelity. So in this case, we chose this one as the best one to move forward. And next, we looked into different starch concentrations. So here you see without ethyl cellulose or lutein, the samples, right? And then we looked into different concentrations of ethyl cellulose from 10 to 6. And this was relatively better 3D printed compared to the others. And then we used that concentration to study further the effect of temperature and starch concentration during printing. So here you have starch concentration 9% and different temperatures. As you see, all the images, they don't look good. Lutein is released and the structure is broken down. So then we went to starch concentration 10. So if you look at especially this sample here, this is one of the best samples we had. As you see here, we don't see really the yellowish color because it was loaded in the center of the gel. But when we increase the concentration further to 11, it doesn't get better, and 12, it was not really printable. So in all those examples, we are using ethyl cellulose as the core. So next, we used Zane as core because this also can solubilize lutein. And here we have Zane 20%. As you see, none of them look good, right? The lutein is released from all the samples. And then when we increase the Zane concentration to 40, now it is able to hold the shape, and further increase did not really help. And in this case, we chose this 14 millimeter per second speed at 40% Zane as the best condition. So whenever we do 3D printing, we always look into quantification of that sample, and that's how we do it. We look into 3D printability, and sometimes we use 2D area or line thickness or height. In this case, we use the volume of the 3D printed object, and we compared it with the model design. So here you see this sample, for example, was insignificant from those two, and this is, I think, one of the best ones we obtained from the image. So by looking at the sample, you have an idea, but it is always good to quantify that and be able to do comparison so that you choose the best 3D printed sample. So once it's 3D printed, we looked into the microstructure, and here we have ethyl cellulose with starch sample printed at 55 degrees Celsius. And under SEM, once it's freeze dried, we see the center here representing ethyl cellulose, and then you have starch in the surrounding area. With starch, interestingly, we are seeing some particulates around. And same thing when we do ethyl cellulose, we still see those small particles. However, when we are using just pure starch, or you look at the outer shell of the starch, we do not see those particles. This suggests that those could be our lutein particles precipitated in the matrix as it interacted with the aqueous water phase. And then we looked into crystallinity. We see a reduction in lutein crystallinity. We looked at FTIR for chemical interactions and DSC for the melting point of lutein. And probably one of the most important data we have is on the storage stability of lutein. So we did storage stability at 25 degrees Celsius over 21 days. And here you see the control is the blue line. And then 3 samples, they are all 3D printed samples. But the difference between them, their shape fidelity and printability are different. So we wanted to see if we just randomly, print, even though the shape fidelity is not good, will that still provide some advantage in terms of its chemical stability? But here we see really differences. It doesn't tell us that if you just do 3D printing, it provides increasement, especially looking at this sample, so that the shape fidelity is really critical in obtaining this significant enhancement in the storage stability. We also did the same test under 50 degrees Celsius. And again, we see the improved stability after it's 3D printed. And the first 2, they were with ethyl cellulose as core. And then when we use Zane, we obtained similar data where the stability of lutein was significantly improved after encapsulation using 3D printing. So the next one we looked into is bioaccessibility of lutein. And in this case, we followed, a consensus paper where we use oral phase, gastric, and intestinal phases with simulated fluids as well as enzymes. And eventually, especially when we did with Zane, we see significant improvement in the bioaccessibility. But interestingly, ethyl cellulose did not release, and now we are looking for different applications where we don't want to release compounds in the intestinal phase. So the last part I would like to mention is on upcycling imperfect vegetables using 3D printing. And as you may know, food loss and waste is a big issue, and we are losing almost 30% of the food we generate. And if you look at especially fresh fruits and vegetables, this number is even higher. It's almost 50% of all the production we really waste. And in this case, we looked into, can we use 3D printing to upcycle those imperfect vegetables? In this case, we used carrot and broccoli. Then with blanching, we freeze dried them, grind them so that we have a powder that is really stable with very long shelf life that can be used for 3D printing on demand. And this is the shape we used for 3D printing. So just basic information about the composition of them. So we use just wheat flour with very basic formulation to do 25% carrot, broccoli, and their mix, and then we did 50% and 75%. So here are the images you see of the 3D printed samples. So again, those are carrot. Here in the middle, we have broccoli, and this is the mix of broccoli and carrot. And this was without any vegetables. And this row represents 25% vegetable substitution with the flour. This is 50%, and then you have 75%. And that's basically supported by the color. You can tell we are using more carrot here, for example. And we optimized this process, and then we did also, bake them in the oven. Then we looked at the texture of those samples, and we also looked into their 3D printability. Again, in this case, we used the volume of the object and compared the 3D design with our 3D printed samples. And here we can see that, for example, this sample and those, they have similar volumes that during after printing, basically. But it's always also good to compare those best samples and look how they are visually looking so that they match with those values. Because you don't want to have the same volume, but a different object, right? So that's also a possibility. That's why you have to do the confirmation as well. So overall, so far, I think we are able to provide, maybe for the first time, showing 3D printing can create biopolymer gel beads. CO2 can really protect the nanopore structure during drying and creates high surface area, and encapsulation of lutein into ethyl cellulose and Zane matrix along with starch gel significantly improved the chemical stability. So in this case, we are seeing improvement from, let's say, 24 to 70%. At 50 degrees, we are seeing increased from 10% to 48% retention after 21 days. And also the bioaccessibility was also improved about 7-fold when you use Zane, especially as the core material during 3D printing. So as an outcome and impact of this project or our program in general, especially on the 3D printing side, so we are able to create spherical bits, and at the same time, we are eliminating the use of surfactants, organic solvents, and you do not need additional purification and separation steps. And 3D printing really can provide precise control over the shape, the size, and composition of the encapsulant, as well as if you are, looking into different composition for delivery purposes, it can provide that as an advantage. It's not only creating a 3D shape, but also it's providing advantages such as improved stability and bioaccessibility of bioactive compounds. And I do think this is one of the first steps that we are taking towards more like personalized nutrition that will enable to do more custom manufacturing of foods. And 3D printing can help reduce food loss and waste, as we did only a few studies so far in that area, but we are looking into extending our research in that field. So with that, I would like to thank my team, Food Engineering for Health Lab, all my postdocs and students, as well as funding agencies for supporting our research. So with that, I will thank you all for your attention, and I will take any questions you may have.
Thank you, Dr. Ubeytogullari. If anyone has any questions, please feel free to come off mute and ask your question. There's a question in the chat that says, thinking of cost of scaling to large volumes typical in the food industry, what type of product can be feasible from cost perspective? Would it be a supplement?
Thank you for this question. Really, we always get this question about novel food processing technologies. And one thing I always like to say, 3D printing of food is relatively new. If you look into plastics or metals, for example, they really made significant improvements, and now they are able to produce mass scale. They are producing car parts and all those things. So in terms of the speed, I think that's the most lacking part. But I think for the cost, if you compare the cost of the equipment to an equipment or systems we use already in a food production facility, I don't see a big difference there. But of course, the speed of the 3D printing needs to be improved. We do see now some industrial applications of 3D food printing, especially on the chocolate side. So I think it's a growing field, and I do think the printers that we use today will get better in the future, able to support the demand, I would say.
Great, thank you. And there's another question from the chat. Would you consider a 3D printed vegetable as ultra processed or fresh, or some, this is me adding to it, somewhere in between?
Yeah, I mean, that's a very good question. And it is very relevant these days, especially we hear a lot of discussions about ultra processed foods, right? So I don't really think that should be categorized in that ultra processed category, but I have questions about, in general, how do you categorize a food in that category? So that's a different question. That is a bigger question. But for 3D printing, what it does, basically, the technology is more like an extrusion. So it's not very complicated, first of all. It doesn't get involved any additional chemicals or anything like that you would not use in a regular formulation. So it doesn't bring any additional ingredients just for 3D printing. So from that standpoint, I don't see 3D printing as just for the processing itself, moving that produce to another category. But also we need to think about those fresh fruits and vegetables. Their shelf life is very short, 1 week or 2, right? And especially the ones we worked on, those imperfect vegetables, the companies, they just throw them away if they do not even fit in the package. So, and we need to think about what can we do with them? Can we really find a solution in that 1 week and make them, reach the consumers? So what we are doing, we are really enhancing the shelf life, and we have not done any nutrition study on them, but I do think we should have similar nutritional composition with those vegetables. But thank you for the question.
Sure, that's really interesting. Changing gears a little bit to more of the bioavailability portion of the work you did. Have you seen that with increased stability, which I know is good for shelf life, is there any difference in the bioavailability? Like, is better, more stable compounds more bioavailable or less, or any correlation?
I think it is. So when you look into bioavailability, it has a lot of different, so basically it builds on everything you do, and then you consume that product. So there are a lot of things that happens before you're able to assess the bioavailability. So that's, from that standpoint, the stability of the compound, will definitely impact the bioavailability, because if it is not stable, you already lost the product on the shelf or the bioactive compound, and that will not contribute to the absorption in the body. We are currently doing some cell culture work to see the absorption, but from just our in vitro simulated digestions, we see the bioaccessible fraction having more of those bioactive compounds, meaning that we have more bioactive compound that is in contact with epithelial cells in the body. So, the theory is we will have more absorption just because we have higher concentration. But I think that's something we are currently looking into. Yeah.
Great. Thank you so much. Is there any additional questions from the audience? Well, if not, I'd like to thank you, Ali, once again. It was a great presentation learning more about 3D printed foods and bioavailability. I'd like to now just let everyone know about the next portion in our series. Our next webinar will be held on Tuesday, October 22nd. So next month, and we'll have two speakers, Dr. Tim Duncan, a research scientist with the U.S. Food and Drug Administration, and Dr. Scott Coffin, who is a research scientist at the California Office of Environmental Health Hazard Assessment. And they'll be speaking on microplastics and nanoplastics. Thank you to Dr. Connie Weaver, the Distinguished Professor of Research at San Diego State University, and who was our 2023-2024 chair of the Phi Tau Sigma Future Directions Committee, for putting this webinar together next month. As we move forward, the registration link can be found in our Phi Tau Sigma newsletter and on the Phi Tau Sigma website. If you have any ideas for future programming, please reach out to Fan Bin Kong, who is the chair of the program committee for the upcoming year. So if there's nothing else, I'd like to once again thank Dr. Ali and thank everyone for your participation in today's webinar. Have a wonderful afternoon.
Thank you all. It was a great honor to present.